Good morning, everyone, or good afternoon, wherever you may be joining us today. We'd like to thank you for joining us on the Nelson Labs webinar today and the topic of impact of device changes on biocompatibility. Now, before we get started, we just have a few housekeeping items we'd like to go through, and then we'll turn the time over to Audrey. If you missed our webinar or would like to refer back to this webinar or any other of our webinars, you can always find them on the Nelson Labs website on the on-demand webinars page listed under Education. You can receive notifications of upcoming live and on-demand webinars by liking Nelson Labs on Facebook or following us on LinkedIn. We'd also like to invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There you will find curated content from industry thought leaders, everything from introductory sterilization and lab testing topics to advanced in-depth learnings from our expert advisors have filled this academy with cutting-edge education, content, and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve patient outcomes and minimize risk. So let's get started. Today we're joined by Audrey Turley, Director of Expert Advisory Services. Audrey has over 25 years of experience working in research, laboratory, and test design function in the medical device industry. She is a biocompatibility expert, having spent 15 years in the lab performing all the in vitro tests offered at Nelson Labs, which include cytotoxicity, hemolysis, and genotoxicity testing. Audrey was responsible for the development of the in vitro mammalian chromosome aberration assay at Nelson Labs. She was also on the team at Nelson, which developed and validated the in vitro irritation assay for medical devices. Audrey is an active committee member of several working groups with the Association for the Advancement of Medical Instrumentation and on the International Standards Organization level as well. So without any further ado, I'd like to turn the time over to Audrey to get started.
Thank you, Mike, for that introduction. I'm happy to be with everyone today. To get us started, I wanted to go through, just a quick outline of our discussion. We're really going to focus on a risk-based approach to handle these changes, and that includes an application of the information we find in ISO 10993-1. And then I want to spend some time talking about what constitutes a change. I think a lot of times manufacturers may not control their full process, and changes may occur that we're not always aware of, but it's important to understand what changes affect and impact your biocompatibility of a device. And of course, we're going to spend a hefty amount of time reviewing some case studies today. So first, I want to start off with looking at our overall perspective in the industry, where we do want to make sure we're focused on a risk-based approach. So this is the title for ISO 10993 Part 1, which is our governing document for biocompatibility. And you can see that any evaluation of medical devices for a biological evaluation starts with within a risk management process. And then just as an example, I've included a guidance document from the FDA. Several other countries have these guidance documents and interpretations of the biocompatibility standards. So for just discussion's sake today, I've included this from the FDA. They have a full section on the risk management approach that we can use in biocompatibility evaluations. So while this slide is quite wordy, I've highlighted some of the important points that the FDA identifies for us so we know what to look at and look for when we're evaluating our devices. And to make sure we're starting on the same page today, it's good to have a really good definition of risk in your mind. So anyone who's ever heard me present, I love to put in definitions. It just helps put everybody on the same playing field. So I always like to pull in ISO 14971 because that is our document that says, when you're looking at med devices, do a risk-based approach for any assessment of med devices. And that document defines risk as the combination of the probability of occurrence of harm and the severity of that harm. And then we can turn to ISO 10993 Part 1, which identifies biological risk very similarly as a combination of the probability of harm to health occurring as a result of adverse reactions associated with medical device or material interactions and the severity of that harm. So in both definitions, it's the probability of the harm occurring and the severity of that harm if it does occur. So I want to make sure we highlight our risk factors that we're going to be focusing on today. When we look at medical devices and we want to address them from a biocompatibility standpoint, we really look at the manufacturing and then the device use. So focusing on manufacturing, the points we are interested in are the materials used, the processes performed, and the location of patient contact of the device. While we also care about where a device is manufactured as far as geographical location, when we talk about device changes, being specific to where that location of change is happening is very important, and you will see that today in our discussions. For device use, we look at patient population, contact type, and contact duration. To further support our evaluation of device use, we turn to Table A1 from ISO 10993 Part 1. You can see that this whole table is based on assessing the risk to the patient based on use. So our first three columns go over general categories of contact, then specific contact, and then contact duration. And this then will indicate what endpoints we need to evaluate, but those endpoints are really the risks to the patient. So we'll use those again in our evaluation of device safety for changes. This table is quite long, so it goes on to 2 pages, but it's very in-depth and very helpful. We're going to use this information, specifically the endpoints, to guide our risk assessment discussions today. So now speaking about what constitutes a change. So ISO 10993 Part 1 states that the biological risk assessment of materials or final products shall be reevaluated if any of the following occur. And I've summarized the changes here to the right. Any change in the source or in the specification of the materials, any change in the formulation, processing, primary packaging, or sterilization, or any change in intended use of the product. So this in general gives us a guidance of what is a change. And then in our case studies today, we'll give specific examples of changes to help identify, is this a change and does it impact our patient population? For evaluating those risk factors, we have some tools that we can use. So I put up this figure from 10993 Part 1. It's what guides us in our initial evaluation of medical devices. I think it's still helpful when we go through these questions for a change, because possibly we already have some data on the materials that we might be changing to. So this is a good resource to start with to understand, do we need to step into testing? Do we need some data? This is a good series of questions to start with. And then I listed some tools that we typically use when we're looking at device changes. Chemistry testing with a toxicological risk assessment, we'll use that a lot. Biological testing, of course. A literature review or comparison to a legally marketed comparator device. Now, this is not a finite list of tools. This is just the more common options that we'll use that I wanted to list here for you today. And then our approach. So this is just to give you an outline of how I will walk through a biological risk assessment. My very first point of focus is going to be on device information. I would say for our team, this is the area that is usually the weakest explained to us from any manufacturer that comes to us, because there's so much concern about the back end. Are we going to have to do more testing? How many devices do I have to make? And there's a lot of concern and drive to get to that end point. However, if we don't spend enough time up front, we could be making some hasty decisions that really don't address our change and really connect all the pieces together. So you'll hear me repeat this often today, and hopefully I'll be able to show some examples to highlight the importance here. When we talk about device information, we really need to understand from a biocompatibility perspective how the device is used, what's the patient population, how many devices are used, and the exact contact time. So we'll go through that in our examples, but that's a really important place to spend some focused time up front because it will make the rest of your decisions much easier in the process. Next, we'll talk about change details. Now, this is where it can get pretty focused, and you'll see that again in the case studies today. Where getting the details very specifically, again, can help you really assess if you need to do more testing and to understand the impact of that change overall. So change details like understanding your new material information, really being specific about the device, the location on the device where the change is happening, and then compare the change device to the previous device to really understand how much is changing and what's the actual risk to the patient. Then we'll evaluate our risk factors, and our risk factors, again, will correlate very closely to the ISO 10993-1 table A1, a lot of ones in there, but we'll focus mainly on those biological endpoints that we've pointed out, using the information we got about our change to guide us in our decision if we need to gather more data or if we have enough information to actually justify that this is pretty low impact and we don't anticipate any adverse response by the patients. Okay, so let's get to the meat of our presentations, which is going to be our case studies. It's where we're going to apply all of this information I've just given at the first of the presentation. I want to first state that the devices pictured in all the case studies are free images that were found on the internet, and they are example devices. They may not match our exact situation, but it's just to give you an idea of patient use, which is important for us to understand in our risk assessment process. And then also that I've taken examples of changes across a broad spectrum of devices and combined them to create new and different scenarios for us to talk about today. So our first device is going to be a stapler. So this is a delivery device that will deliver staples during a surgery. So it's used to staple the tissue during surgery. Exposure time is typically 5 minutes. So I just want to point out here that we're getting very specific on the intended use, where we know from ISO 10993 Part 1 in that table that we care about being less than 24 hours. That's our general category, but really highlighting that it's a very minimal time within that 24-hour window that this is used is really important so we can understand true risk to the patient. Our device categorization is external communicating limited with tissue contact. Our change is that we are moving to a new color house. So again, remember this is an example device. Our actual device does have a color on it. So we're moving to a new color house. What's our impacted portion of the device? So we're going to focus on our area first. So the patient contacting portion has black, gray, and purple components. And purple is the only color that is changing. That's the only part of the color house that we had from this new supplier that we had to change. The purple portion of the device is 5 centimeters squared. And our full patient contacting portion is 134 centimeters squared. So don't worry about the math yet, but you should be able to tell that our purple portion that's changing is a very small percentage of the actual device that's contacting the patient. Further details on our colorant is that the colorant is only added at 1% to the base material, and the base material remains unchanged. So again, it's only the colorant added at 1% to that 5 centimeters squared. So again, really detailed in the changes, but hopefully you'll see how this plays out to really support your assessment. So we're going to go through the impact first. So our risk factors that we're concerned about are based on our biological endpoints: cytotoxicity, sensitization, irritation, acute systemic toxicity, and material-mediated pyrogenicity. So these are the endpoints identified because of our patient contact, which is external communicating, limited tissue contact. Based on the amount of color change that we evaluated in the previous slide, and I've done some calculations here in the blue box at the bottom of the screen, we can see that a very small percentage of the device is actually changing. So our surface area is really only 3.7%. And when we take into consideration that only 1% of that surface area is changing, then it's 0.37% of the device has the change. So we want to keep that in mind. The device has a short contact duration. So even though it's limited, we're very specific here again, and it's 5 minutes. And we also understand that the colorant is added to the device in a form that is not expected to leave the base material. So it's manufactured in such a way to be solid. Now that doesn't mean that toxins couldn't leach out of the device or that material. However, it is not designed for that intent, so it's expected to stay intact, especially for a short contact duration of 5 minutes. So when we go to our decision line here, we're definitely going to write everything up in a risk assessment so we can have documentation of how we assess this change. Next, I'm going to think about, is this amount of device change an impact to my patient safety? How can I gauge that? Well, I know that 0.037% has changed. And I also know in ISO 10093 Part 12, which guides my sample preparation for biocompatibility tests, allows me to have a plus or minus 10% margin on surface area calculations for my testing. That means that there's this buffer in the testing of 10% when we calculate the surface area and determine, how sensitive our tests are here. So I think to myself, okay, with a 0.037% change, is that significant enough that would make me think I need to repeat some testing because we would be able to pick up this change in the extracts? So in my experience, I would use this to say, we know it's an insignificant amount of change. So then we would look and see, okay, is there a risk from the colorant information? So then I'm building my discussion, right? One thing on top of another. So I talk about how insignificant the change is because it's so small and I have data and literature to back that up. Then I'm going to go talk about the new colorant information, put in everything I have from the color supplier, and research my toxicology info on the colorant to understand if it's toxic, and also speak to the the colorant not expected to leave that base material. So I'm going to use all of those things in my decision line here to write up a solid risk assessment to assess patient safety. If I wanted to really seal this deal, I could also do some cytotoxicity testing just on some purple components without doing the full thing to just understand, hey, is there any indication that there's a toxic substance that's leaching from here that would need further assessment that maybe I might be concerned about? But that's how I would approach it for this type of change. So that's one example how this being very specific about our change, really understanding down to the details, very minute, and that can support a really strong risk assessment discussion. Okay, case study number 2. So we're going to talk about catheters for this case study. These are used for revascularization. I didn't put it in here, but I'm going to say that the time of use is less than 3 hours for the procedure. Our categorization is external communicating, circulating blood with limited contact. And our change is that we are moving to a new EO ethylene oxide, so EO sterilizer, which includes a change in cycle parameters. So I wanted to get an outline of what's different in the cycles. So I have received this comparator table, and we can see that from the current cycle to the new cycle, every single parameter is slightly different. So I step back for a minute and think, okay, from a biocompatibility perspective, it's difficult for me to, not just me, but difficult to find data to say, how does the relative humidity impact our EO sterilization impact on the device specifically? But we need to also take into consideration the temperature, the initial vacuum, humidification, all the different parameters. So it's everything that's changing just a little bit. And so how can we assess this for biocompatibility when everything is changing? And you may be thinking, I thought I joined a biocompatibility seminar, not a sterilization seminar. But remember that one of our risk factors that we need to look at is processing. And we know that EO can have a biocompatibility impact. So that's why we want to assess this, not only for, you would need to repeat some of your sterilization validation work to approve this next supplier, but we also have to think about any impact from the biocompatibility portion to the patient. So we're going to look at our impact line, and here again, we'll start with our biological endpoints, which are driven, again, not by the change, but by the use of the device. We're going to start there first. So we have cytotoxicity, sensitization, irritation, acute systemic toxicity. We have MMP again, and the new one here is hemocompatibility because of the blood contact. So the first thing that I would check as a risk assessor is I'm just going to double check the material compatibility with EO. So I would ask for a materials list of the device, and then I would partner with some of my sterilization experts and say, let's just give this a once over to make sure we've double checked that this new cycle is not going to impact our, material or our function of the device. Then I'm also going to note to myself, well, EO and ECH, so ethylene chloride, these are the residuals from an EO process. They are known sensitizers. So again, we can see that the change that we're going through, we can directly correlate possible impact back to our endpoints. So we're going to look and see how can we address those sensitizers. So in my decision line, I really want to see the EO and ECH residual data for the new cycle. And I want to make sure that that data shows acceptable limits for ISO 10993 Part 7. So this is an interesting point where sterilization and biocompatibility overlap. So for EO sterilization, we have a residuals document that resides with the biocompatibility expert working group, because we have the toxicologists to assess whether those chemicals would be toxic to patients based on device use and patient population. So again, that data is really going to tell me, okay, we're only worried about the change in sterilization. We're focused on that change. What risk does that present? Well, it presents we could have residuals that would cause a sensitization response. So I'm going to then document everything in a risk assessment, and I'm going to connect that residual data to the risk of the device. So one thing I see often too is that a lot of times we just want to get the data and send it to our reviewers and say, okay, everything's fine, but they want to see us connecting all that data back to the risk of the device use and to the patient population. And a lot of times, of course, we think of EO residual data for sterilization cycles, not necessarily for biocompatibility. Even though they tie together quite nicely, we just need to add that into the discussion. And so this is how we would approach a change in sterilizers for EO, just an example there. Okay, case study number 3. I just want to give a shout out to my daughter. So it's really hard sometimes to find free pictures of devices. So this is my daughter's dental aligner. She let me take a picture of it. So shout out to her for being part of our webinar today. But this is our example is a dental aligner worn daily when not eating for approximately 24 months. And our patient population extends from adults to pediatric patients. Our device categorization is external communicating tissue bone dentin long-term. Now, some of you who are possibly biocomp experts could be shouting right now at your screen saying, wait, this could also be a surface device, mucosal membrane contacting long-term. And you're absolutely right. A device like this can fit into multiple categories. So ISO 10993 helps us with this scenario and says that if a device can fit into 2 categories, you pick the riskier, the higher up category. So if we really look at the mucosal membrane category contact for long-term devices, and we compare the biological endpoints that are of concern to those for external communicating tissue long-term contacting devices, they're actually almost identical. The only difference is the requirement for a material-mediated pyrogen test in the external communicating category. But if you actually look at some of the other government regulatory bodies like FDA, they actually want you to include the material-mediated pyrogenicity in there anyways. So very similar, and I picked the escalated category, which is our external communicating tissue bone dentin long-term category. So just an example of how we can settle some of those disputes to make sure we start on a clear path for our risk assessment. Our change for this device is we're moving from one company for our base material, which is a TPU, to another company for another thermoplastic polyurethane. So information from company A. So we've already done biocompatibility testing on this TPU. So it was our original material. We did all of our testing on that and passed. And our manufacturing process from the beginning has been thermoforming. Company B, though, this new TPU, has completed some biocompatibility on their material, which is cytotoxicity, acute systemic toxicity, and implantation. So some of you may recognize this as USP Class 6 testing. And the manufacturing process, though, is a little bit different for this new material. It's thermoforming, but you need a release agent. Another important thing to note here is that formulation for each TPU is not available. So one thing to note is that one supplier's material is not the same as a second supplier, and we have a really clear indicator of that here because they're not giving the formulation. So when you think I'm just getting the same material from a different company, if they don't share their formulation, it tells you that they have proprietary additives and processes that they use to make this material. And so when we give these material names like TPU, it's more of a, hey, it fits in this category, but of course there's freedom to manipulate and make the material fit the needs of the industry they're providing to. So just one thing to keep in mind as we go through our rest of our risk assessment here. So again, we're going to start with impact. We've listed our biological endpoints, and it's everything except for hemocompatibility. So you can see there's a lot of, we have the chronic systemic toxicity, genotoxicity, carcinogenicity. These are the long-term endpoints that we're concerned about when we have a device that has long-term patient contact. I also want to make sure I keep in my mind as I go through this that we have a pediatric patient population that's involved, so that creates a higher risk because they're still developing. And our greatest risk here for this product is honestly the leachables over long-term exposure. So I say greatest risk because, again, we're going to think about our definition for risk, right? Where it was the probability of that harm occurring. So this is a TPO, it's in the mouth all the time. We know we're going to have leachables coming out, and it's the impact of that risk. So when we think about long-term exposures, we think about a chronic toxicity response, carcinogenicity. These are high risk endpoints that we want to make sure we're evaluating carefully. So when I go to my decision point, I have to realize that company B, I'm going to just go back one slide, their testing doesn't address long-term exposure. We can see here we have cytotoxicity, acute systemic toxicity, and implantation. So it followed USP Class 6. That means that my implantation test was about 7 days. So again, not very long for me to understand a long-term exposure. So with that in mind, I think I'm going to run a cytotoxicity test just to give me some initial data to let me know, are we seeing toxic leachables come off of this product? I'm going to do some research on what I can get from the supplier on that TPU material, the new one, to see if, man, do we need to step into sensitization or irritation, anything from that short-term exposure? Now, remember, those are risks, but the severity, you know, you can just remove the mouthpiece and those responses will go away and they can change to a different material. So this isn't a life-saving device or anything like that, and it can be removed quite easily. So I would say the severity of the harm from one of those two is pretty minimal. I'm most focused on these leachables that are going to happen over time. So I'm going to step into my first option, which I really want to pan out, is I'm going to reach out to my supplier from company B and ask, hey, do you guys have other customers that would let me say that this is, used for their devices that has this TPU and uses that same release agent? So you can have this legally marketed comparator device discussion in your risk assessment. If that doesn't pan out very well, my next option is, honestly, we need to step in to gather data on this long-term information. So I'm going to run some chemistry and I'm going to do a toxicological risk assessment. And that may seem, I want to be careful here because we do strive to assess this without, using all the information we have available, but we do have to be willing to acknowledge when we don't have the long-term data on a specific material. And this, even though it's a TPU used in the industry, we don't have it from the supplier, and we have to be protective of that pediatric patient population. So when I work out option 1 or 2, whatever one is working out, I'm going to write a risk assessment. I'm going to connect my data to my biological endpoints, and I'm going to include literature review of that release agent because that's the new information. Normally we can, we'll know specifically what that is so we can put more detail in our document and tie everything back to my endpoints. So one thing I want to point out, and this is a perfect situation here, is there could be a third or a fourth option. That's the unique thing about risk assessments and assessing device change, is that it really depends on specific history of the device. There could be 5 other points we could look at here, but for our discussion today, I needed to keep it a little more focused. That I understand that the bigger picture can be complicated. The goal of these case studies is to help everyone understand this is how I can walk through it, how I can ask these questions, and just thinking outside the box a little bit and using our, more of our critical thinking skills to do the risk-based approach. Okay, so we're going to step into our last case study for today. We're going to evaluate a polymeric implant family. So there's several of these components made of this material. This is just an example. This one's used for joint with long-term tissue bone contact. The change we're focused on here is in the process. So they added an IPA rinse at the end of processing just prior to packaging. So we can review that change. Not a lot to review here, but you might say, why would you add or change your process here? But the purpose was to reduce their bioburden. So they wanted to add this rinse to have a cleaner product at the end of manufacturing. So I think this is interesting for us to think about changes like this, because we may be focused on improving one test outcome like bioburden for your sterilization. So you could have maybe a lower sterilization dose, which might help the integrity of the product, right? So we're all for reducing that dose, but that change intended to only impact one thing now impacts biocompatibility. We want to make sure we assess that carefully. Okay, so we go through our same process. We're going to look at our impact. Again, this is all the biological endpoints, everything except for hemocompatibility. But from IPA, it's kind of a short-term response, which is what we normally think of short-term, which would be irritating, but this device can't easily be removed if it irritates the tissues. And we can have acute toxic poisoning if there's too much residual. So we want to just be thoughtful of any discomfort we could cause the patient. So we definitely don't want residual IPA left on the product. Since we know that this is the only thing that we're changing in the process, we're really going to focus on IPA specifically. So I feel like we would want some more data here. We just want to see if there's some IPA that's residual hanging out that would create a biological response. So I'm just going to quickly run a cytotoxicity test to see what the impact could be. We did this, and our IPA, or the testing failed, showing that IPA is still present. We know this because a device without the extra rinse passed, and this device failed. So that's how we can isolate that it's the IPA. So we decided, let's go ahead and do another rinse after the IPA, and let's see if that gets rid of the residual IPA. So the rinse was performed, and we went ahead and did another cytotoxicity test, and the device now passed. So pretty simple process, but this is a way that you can use cytotoxicity to kind of guide a risk assessment. It's a relatively inexpensive and quick test, especially when we're talking about a low risk chemical in this situation, which even the residual level on the device was most likely very low, but cytotoxicity testing is so sensitive that we can get a good indicator if there's further assessment needed, or if we need to get rid of the IPA rinse and come up with another strategy to reduce bioburden. But this is the process we would go through. And again, capturing all of this in a risk assessment, focusing on the change, indicating what risk that change presents in the context of the biological endpoints, and then being able to state no impact to patient safety at the end with our data. Now that we've gone through a few case studies, I just want to speak to what would be valuable to include in a biological risk assessment. It's important that everything is documented. So even if you determine that the change is low risk, you still have to, write that out so that it's documented, not just for auditing purposes or submission purposes, but also just for that tribal knowledge. I find a lot of times in my customer visits or customer phone calls, I'll be talking about, hey, tell me why you guys do that. And it will be, I don't know, the person before me just did it, so we kept going. It's important to keep these documents in place so that you know why you're doing things, so we don't get in a pattern of testing without understanding and maybe generating data that isn't supportive of the change or what you're trying to show for safety of your product. So again, starting with a really thoughtful device description, we always want to include pictures, and to clarify patient contact, and pictures can be helpful with that as well. I would say it's important to remember your audience for these documents will rarely be an expert on your device alone. They might be an expert in the realm or category of devices, but maybe not specifically your device. So make sure you have good device description, and go into how many devices are used or possible with the patient, and then the specific contact duration. One thing that's important when we talk about contact duration is cumulative use. So a really good example of this is like a continuous glucose monitor. So those are used from 10 to 14 days, but then they're changed out and they're used for the lifetime of the patient. That would be our worst case scenario, right? So we wouldn't want to say that the device is only used 14 days because it's repeated. So every time we apply a new device, the patient then gets a fresh dose of whatever was coming off the device. So we like to change devices because that's good from a cleanliness perspective. But we just want to make sure we're taking that into consideration from a biocompatibility perspective, that we think about the long-term contact. Okay, and then talking about device categorization, I always stress that we should follow the language that's in ISO 10993-1, and try to remove any creative opportunities here in categorizing the device. Just use the strict categories that are listed in 10993 Part 1, and be careful of using direct versus indirect contact. There are times that that's an important conversation to have, especially for device description and device use. But for device categorization, there's actually only one category that uses the term indirect, and that's for external communicating devices with blood path indirect contact. And that greatly impacts how we assess hemocompatibility for those devices. So that's a critical indicator. For all other devices, we just want to stick to the terminology in 10993 Part 1. It helps because it puts everyone on the same page, and that terminology has already been vetted out to translate across all the countries that use the document. So it helps in your document review. Change details. Again, you can see from the examples we use, be very specific. I recommend to use tables when you're outlining the change. Just make the information very digestible and clear so that when you go into your risk discussion, it's a very focused conversation and we're not getting distracted with other details of the change. And then in the risk discussion, report your data, do your literature research, and make sure when you're reporting data or and putting out your literature research, tie that back to the risk every time. You know, this is like crossing your T's and dotting your I's. Just make sure everything ties together. And then, of course, at conclusion, summarize and repeat your previous statements. And just in summary, I wanted to bring up this slide and one more to just remind you of our risk factors and what we want to look at and stay focused on when we assess changes for devices and their impact to biocompatibility. So we want to make sure we're looking at the manufacturing side and correlating that to the device use. And again, just an outline of the approach, the flow that we should use in our discussion for our risk assessments. And if anyone in the audience today finds that you're in a situation where you could use some help, maybe the change is a bit above and beyond what you've handled before, we have a full team, including myself, to help you with biocompatibility needs. So we have Dr. Fragel, Hurst, and Lauer available, and our project manager, Darcy, who helps keep everything going and on track. But you can certainly reach out to our full team at advisory services at nelsonlabs.com. And one last thing, I just want to tie in, Mike mentioned earlier about our Sotera Health Academy. If you find that you'd like to build your expertise in biocompatibility, and of course, any of the other areas that are related to medical device testing, you can join our academy. It's free to register, and you can just browse our content. We will start adding paid for content later. And it'd be good to be familiar with the academy, and then you can learn to see which courses you'd like to sign up for. And I think it's a great avenue for you to bring your team in to learn the information that you're engaged into to help them perform their jobs better. So thank you for joining us today, and I'm going to turn the time back over to Mike.
Well, thank you, Audrey, for those great insights today. I think we've all learned a lot of information about changes, about biocompatibility. So we do have a lot of questions, and we will try to get to as many as we possibly can in the remaining time that we have today. And if Audrey isn't able to get to your specific question, I'm sure she'd be happy to address it individually after we conclude today. So let's start off with the first one. Okay, here we go, Audrey. So how should the form of the risk management report in implementing ISO 14971-2019 be related to the implementation of ISO 10993-1? Do they have to be separate?
Yeah, I think that's a great question. So one thing to remember is the ISO 14971 document is more of a broad, risk-based approach to medical devices. So when we assess risk assessment, or when we perform risk assessments, especially on changes, we cross back and forth between both documents, but we stick mainly to ISO 10993 Part 1. That is because that document, and the 10993 Part 1 specifically addresses our area of focus. So we want to talk about biocompatibility in the context of biocompatibility, and 10993 gives us that guidance. However, we do bring in rationale for the overall approach from 14971. So we'll reference, like, when you do risk-based approach, the idea is to use the data you already have available. So we'll pull quotes like that from 14971 to support our evaluation that we're doing under 10993 Part 1. So I know that's not very specific because it's more about, following the methodology, but it is followed in that way. But we want to use from 14971, but we want to use rationale that makes sense from a biocompatibility perspective, which is specifically looking at the endpoints, the testing available, the approach we would use, like cytotoxicity or doing chemistry, those type of tools fall under 10993 Part 1.
Okay, great, thank you. Here's our next question. Can we justify biocompatibility for the limited contacting devices if the vendor has a substandard cleaning validation?
Was it really substandard or did it say substantial, Mike, on that question?
It did say substandard, but maybe I'm guessing substantial is probably intended.
Yeah. Okay, so we love to use cleaning validation data for our short-term contact devices just because it does speak to, if we already know, materials are not a biocompatibility risk, we're really focused on processing. So this is perfect for like titanium implants, for example, where we have a lot of information on titanium. We know the safety, but we don't know about the process. So cleaning validations can speak to low-level detergent or residuals from that process at a level that has been determined to be safe from a systemic standpoint. So we can use that, again, as long as you tie the cleaning validation back to the endpoints. We have had some feedback that that's not a preferred pathway. It's very, very rare, but I think it was actually just missed in the initial conversation to be really clear about how we were using the cleaning validation data, that it's appropriate to look for detergent residuals or oils, or to ensure that your endotoxin levels are low, that we could use all of those endpoints from a cleaning validation standpoint that also will address sensitization or irritation or a pyrogenicity endpoint for biocompatibility as well.
Okay, continuing on, here's our next one. It says, may I ask where, if at all, the concept of biological equivalence is defined in Annex C of ISO 10993-18 fits into the evaluation of the impact of changes on biological safety?
Okay, so biological equivalence, man, that's pulling some definitions I don't have readily available in front of me today. But we want to think about, I can speak to it generally without a reference specifically to -18, which is on our chemistry testing. Biological equivalence would be that we could demonstrate. It's a little tricky because it would almost seem like, oh, you could show that you have all the same responses in your biocompatibility studies for the device, for each device. So that's the only way to really show that would be to actually step into all that testing and gather the data to demonstrate biological equivalence. So I think that is not as common of a used tool for assessing risk or assessing device changes. So I would just caution when we state equivalence overall, that has to be demonstrated line for line. And I know this with the FDA, like they really want to see, like, if you say this material is identical, you have to show that the source is identical, like in a table, lay it out and show that everything is equivalent. So we want to be careful when we use that term equivalence in general. So I would just caution that approach.
Okay. And here's our next question. What if you already have products that are already IPA washed and you are now adding this to another product? Would biocompatibility risk assessment still be necessary?
Yes. When we think about, it's not just the IPA, but it's also the material that we have to be consider. And we also have to think about not just the material, but also the shape and geometry of the devices, if it can be removed as easily from one to the next. So one thing we know can happen is that, especially these cleaning processes we look at, will have suppliers that do manufacturing for several different types of devices, and they'll validate their process based on their worst case device, meaning it has to be considered of material, like cleanliness, ability to clean that material, ease of cleanability, and also geometry. And so you can possibly do like an adoption of a product into a cleaning process like that, but we would want to make sure we're evaluating those factors that can contribute to maybe holding on to that IPA more than another product.
Okay. Our next question. If we author the risk assessment internally, is it still recommended that an expert like somebody from Nelson approve it?
Yeah, so when we think about what is an expert, whatever we're authoring, we just want to make sure we have the expertise to back our signature on that document. So if you're internally authoring a document and you're saying, hey, we have the expertise to assess, this biocompatibility, we understand the data, we understand the literature review that we performed, and we have education and experience to support that, then you're fine to do that. Because we don't really have like a firm definition of what makes an expert in our ISO documents. However, when our CVs are sent to regulatory agencies so they can vet that interest or vet that expertise, they're looking for, what job market you've worked in and your education. So I would just say, make sure if you're signing a document that you really can go to the FDA or to a notified body and speak to what you've written in there. And if you can't do that internally, then you should have an external expert look at it for support.
Okay, continuing on, we have a little bit more time. What is the dependency of the initial risk management file with the risk assessment of these changes mentioned in case study 3? I'll repeat that. What is the dependency of the initial risk management file with the risk assessment of these changes mentioned in case study 3?
Yeah, so I just wanted to flip back to case study 3 so we could all be on the same page. So we're not really using a lot from the initial risk assessment that was done. So if I understand the question right, it's saying how much can we use from the initial risk assessment done on the device, the initial biocompatibility testing. When we look at this aligner, it's full material, it's full, we're changing 100% of this. So there's not a lot we're going to lean on from the first risk assessment. We're really going to look at it and say, okay, what can we get from our supplier for this information? We know they did some initial screening on the material, but we need to address those long-term endpoints. So we're really going from almost starting from scratch here when we look at a different material change for this device.
Okay, our next question. Is chemical characterization mandatory or testing on predicate device which uses the same material manufacturing process sufficient?
Read that to me one more time, Mike.
Yeah. So is chemical characterization mandatory or testing on the predicate device which uses the same material and manufacturing process sufficient?
So I guess it depends on what the change is. So if there's no change, then hopefully we don't, you have all the data that you need. Chemical characterization testing, specifically, like just for this example that we have up, was brought up because that can address our long-term endpoints. That's the preferred route, is to use the extractable leachable collection. We do know it's extensive, and I wouldn't say it's the most expensive. It's definitely cheaper than some of the in vivo options, but it's an investment to step into that. So yeah, I would say that's not our go-to if your materials and processes haven't changed. We try to look for different alternatives. Like you could see, using cytotoxicity or EO residual data, some other options outside of that as well.
Okay, we have time for just a couple more questions. And here's a question that I'm pretty sure you have a great example for. And the question is, how is a manufacturing location change considered? Same product and processes.
Yeah, so this is one thing I said at the beginning, we don't specifically think of location of manufacturing. That's not identified in 10993 as a change, but it's actually a change. So we want to make sure we're considering that. So I would say there should be some evaluation of a change in location of manufacturing. In the example that was given where it's, the same equipment that's used, same materials, we just kind of picked everything up and put it in a different facility. I would say that should be assessed. I do have an example of this. We had a client that did that very same thing, moved from one facility to another. And we said, hey, just run cytotoxicity on your product at the new facility. So they did that and they failed the cytotoxicity test. And we said, okay, that's got to be a fluke. Send in some old product, from the other plant, send in some new, let's up the samples, the sample count for better statistics, let's do it again. So we did that, and the product at the new facility failed again. So we said, well, we better step into some chemistry testing to see what it is. And so we did the chemistry testing, and there was a weird compound coming off, one that we hadn't seen before. And when we researched it, it was a product that was used in surfboard manufacturing. So we reached out to the client and asked them if, told them what we found and said, do you know why this would be on your product? And they said, oh, our neighbors in this new building manufacture surfboards. So there needed to be some work done on the ductwork to clarify the air system so they wouldn't have this fallout on their product. So again, it's a good example of like a change that doesn't seem to be a change, could have an impact on the product. But again, cytotoxicity testing was a good indicator to help us know which direction to try. And then we moved into the chemistry to get more information.
Okay, we'll have time for one more last question here. So what options are there if suppliers, especially colorant suppliers, do not provide information as they deem it confidential?
Yeah, that does present a lot of issue in the industry because of that confidential portion. So we really handle this differently for almost every single product we've come across. We have a few options, but it just depends. Sometimes we can get information from the color house that says, like, we don't have any, and they'll go through like REACH, or they'll list all these standards of toxic hazard chemicals and say, we don't have any of these included. So we can use that for discussion depending on the contact of the device with the patient. We also can sometimes get, like I had in our example today, the percent added to the base material. So we're not asking for any specific formulations, just how much colorant we're putting in that base material. We can talk about that. Sometimes we can run some calculations to talk about potential of leachable through the matrix of the base material. Really just seeing if we can avoid, repeating all the testing, especially if it's a very small percent of colorant that's added to that base material. I'm trying to think of other situations. I have gotten into some legal debates with the color houses for not being willing to give that. We've been willing to sign NDAs where we can sign a non-disclosure agreement with the color house, so we would not disclose their recipe to the client, but we could assess for the toxicology and then keep that as confidential in our file, so it could be reviewed and audited if needed, but it would not be released to the client. So those are just some of the approaches that we've used, and really speaking about the potential of that colorant really leaching out of the device and impacting the patient. That's harder for long-term exposure products, but a lot of times what we find the colorant's an issue in are these short-term devices anyways. So those are just a few examples. I'd be happy to troubleshoot that with you if you wanted to send an email to that advisory services email.
Well, thank you, Audrey. And we have many more questions that we will pass along, but unfortunately, we have ran out of time today. And we would like to thank Audrey for this thorough and informative presentation, and for her insights and industry knowledge on this topic. We'd like to thank you, the viewer, for attending this session, and we hope that you have found this to be a valuable experience, and we hope that you have a great rest of your day.